Good evening, everyone. Okay. It's great to be here, and today I'm going to talk a little bit about the Americas and the settlement of the Americas. And as every other person coming from the Max Planck, I have to start with the typical slides about our institute, how fantastic our institute is. It's in Jena. It has been already introduced to where Jena is, kind of middle Germany, in the center of Germany. And our institute is actually quite recent. It was established in 2014, and the work there began only around in the mid-2015. So it's quite recent, and as you can see, it is composed by two main parts. There is an old villa where the administration is, and also the IT department, and there is this new kind of building with the guest apartments up there. So with a bridge in between, kind of bridging, in fact, the old and the new together. And that's actually what we are trying to do in this institute, the Science of Human History. So a little bit about what is our research profile. We have three directors. One is Russell Gray, that is leading the Department of Linguistic and Cultural Evolution, to which Mattis List is part of, and is going to introduce the department later. Then there is Johannes Krause, that is the Director of Archaeogenetics, which I'm part of. And then we have Nicole Boivin, that she is leading the Archaeology Department. And a little bit about the research profile. What is the main idea is actually to bridge the gaps between humanities and natural sciences. And to do that, we at least try to do some interdisciplinary research of human history, of course develop new methods, and in general study and research on different cultural as well as biological phenomena, and bring this information together to draw some inferences about our past. Okay, now I'm going to introduce what we are doing in the Department of Archaeogenetics, and you have two main lines of research there. The first one that you see there is the studying the genome of historical pathogens. What we usually do, we at least how we started, go into mass graves, mass burials where you have many skeletons buried all together, and extract the DNA from, for example, the teeth of these people, and try to see if we find, beside the human DNA, also the DNA from the causative agent of this of this pathogen, of this death. So why these people die? Which was the bacteria or the virus that killed this person? And then once if we are lucky and we find that, and we try to reconstruct the genome of these pathogens and also reconstruct their evolutionary history. And then, of course, we are really working in the direction of host and pathogen interaction, and see how, both the pathogens and humans co-evolved throughout time. The second really strong focus on our research, I would say the strongest because I'm part of it, but it's equally shared, is the human population genetics. And here what we are doing is extracting DNA from ancient human remains to get and to collect insights into the past population migration as well as admixtures. And we try to link in this kind of multidisciplinary framework the genetic information with archaeological or historical information, and in some cases also with linguistic information. And the main goal is, of course, to try to reconstruct the human population history through time. So just to show you a little bit about which are our facility is kind of CSI kind of work we are doing there. The core of our facilities is so-called clean rooms. They are, and we are really lucky to have available this really large laboratory of 250 square meters. It's the largest ancient DNA laboratory that exists so far, and it's really good actually because okay, it's really a pain because you need to, of course, keep it clean, and the bigger it is, the more cleaning is necessary. But it's really good because this allows us to split our work in different rooms, and in each room we do a different step of the workflow. And in this way, we try to reduce cross-contamination between samples, and also to contaminate with our own DNA the ancient remains that we're working on. And of course, then there is a larger part of our laboratory that is a molecular biology lab where we do all the amplification of the DNA, and then eventually we sequence the DNA on these Illumina sequencer that are really powerful machines that produce million or billion of DNA sequences in few hours. And here, just wanted to show you some pictures, some snapshots of how we work in the lab. These are pictures taken by a journalist from the New York Times. It was completely wrapped up to not only him, but also the camera to enter in the clean room. And that's kind of some ideas. We try to protect not ourselves, but actually the sample from contamination. We wear these suit, glass, goggles, masks, and the only thing that is not real about this picture is usually we actually keep the light on. It's not that we work in the dark, but I guess it was scenic for the UV lights, the contamination. Okay, so what I want to talk today about is this paper that we published last year about reconstructing the deep population history of Central and South America. Really bold title, and of course it's just the beginning of this research. And one of the reasons why I'm actually really happy to be here is that we are really trying to expand further this research in different regions and different temporal horizon. And the settlement of the America is a really long debated question, and so far what we know from the genetics, both modern and ancient genomes from Native Americans, is that the source population of non-Arctic populations, so the population that are excluding the Arctic one that receive later population movement from the Inuit, for example, the Eskimos. But the non-Arctic population of the America derived from one major gene flow event from a population that is formed by two distinct genetic ancestries. One tracing back to an Asian population related, for example, to Han Chinese, and another one, a population that was discovered in Siberia on the Baikal region and has as a proxy an individual hunter-gatherer individual from 24,000 years ago that represent a good proxy for this, let's say, Siberian population that mixed in the past, more than 20,000 years ago, with this East Asian group to form the source of this major migration into the Americas. And as you can see here, once the population, this stream, this group of people managed to actually enter in the Americas, they rapidly split into major groups. One that is here giving rise to the genetic ancestry present in northern Native Americans and Athabascans, and the other one that went down and provided the genetic composition of Central and South Native Americans. The same year of this paper was published, there was another paper that I want to point out where they arrived to the similar conclusion, these two sources to explain the genetic diversity present in the Americas. But the researcher found a signal of yet a third population, a third group in individuals, really isolated groups from Amazonia. This population are Caritiana, Surui, and Chavanti. And those groups, on top of these two ancestry, the let's say the Siberian ancestry and the East Asian ancestry, they have a third component that is shared with populations that are in Austro-Melanesia, so for example Papuan or Ongi, and it has been named population Y. It so far is still not clear how this genetic component arrived and survived through time in this isolated place. So the oldest genome ever published so far from the Americas is this Clovis genome. It's called ANSIC1, and it's a Clovis-associated genomes. Who are those Clovis? Clovis are among the first population or groups present in North America, and they are characterized to be really successful megafaunal hunters, and they are, typologically, they are characterized by these points that are called Clovis points. And what was really exciting about this paper, published in 2014, was that when the genome of this ancient individual was sequenced and compared to modern day individuals from all around the world, it shows the highest genetic similarities to people living today in Central and South America. So placing this genome on this southern branch of expansion into the Americas. Last year was kind of a prolific year for ancient DNA from the Americas, because we finally got more genome from North America, and in this publication in Science showed that they could sequence genome both from the coast, the Californian coast, as well as Mexico, and then also from north and southern southern Ontario, and they could show that these two groups, these individuals fall in two distinct groups, and they actually resemble this early diversification that I was telling you at the beginning between Northern Americans and Central and South Americans. Just to be more neutral in terms of where let's say about the geographical location where the split happened, we just described them as, we just called them ancestral B for the one that, this group that left traces in Northern American population, and ancestral A for people that left traces in modern day people from Central and South America. So we'll talk about ancestral A, ancestral B later on. So as you can see here from this map, unfortunately, there was, I mean, before, by the time of this publication, there was almost no genome, ancient genome published from South America. So we tried to start, and we started a project, we already started a project to actually try to fill these gaps, and with three main questions in our head. We wanted to understand if Central and South American variation of early, in the past, mirror the population structure that we see today. So if there is some genetic continuity through time, we want to understand what is the minimum number of genetic exchange between North and South America. That means how many waves we have from the north to the south. And then we want to, of course, try to reconstruct the relationship between different groups in Central and South America through space and through time. So, and this was the data set that we assembled last year. It's 14 new genomes, sorry, not 14, 49, it would be a little bit less. 49 ancient individuals spanning between 11,000 and 600 BP. But as you can see from our timeline, from the oldest, that is ANSIC, the Clovis-related genome, to the youngest, 35 of the new ancient genome that we report are actually older than 3,000 BP. So we really try to understand what is the diversity of the initial settlement of Central and South America. And those are distinct, divided into four, let's say, geographical transect. We have individual from Belize. Another transect is from the Central Andes. Then the third cluster is from individual from the Southern Cone, so from Chile and Argentina. And finally, we have some individual from the Southeast Brazil. And this map here that you see is not only reporting the new ancient individuals, but also is representing a meaningful statistic. And probably you cannot see really well, but the different colors of these dots and squares represent the affinity of those individuals compared to Clovis. So we wanted to test if every individual in Central and South America is equally related to the oldest genome, the oldest North American genome, that is ANSIC1, or there are differential relationships. And what we were really surprised to see is that actually the two oldest genomes that we sequence from South America, that there is that are population from a group of individuals from Lapa do Santo, dated to around 10,000 years ago, and one single individual from in Chile, in a site called Los Riles, dated to 11,000 years ago. So the oldest genome show a higher affinity to Clovis. So the oldest genome show a highest affinity with the oldest from North America. So we were really interested to study that further. So what we did, first of all, is like typical PCA, principal component analysis, to distinguish population in the in a genetic map. And what we identified clearly is these two clusters, ancestral B, North America, and ancestral A, that contribute to Central and South America. However, if you see where our ancient genome from South America fall, they're all on top of each other. We were not really able to distinguish that based on this principal component analysis. So we decided to use a different technique, that is this F3 outgroup statistic represented through MDS plot. It stands for multi-dimensional scaling plot, and this is actually nothing else than a matrix that measures the dissimilarities between different groups. And what emerged from this analysis is that we could actually group ancient individuals based on their geography. So we could identify a group of individuals both very ancient, but also more recent, from Fuegians, from Fuego Patagonia, forming one cluster. Then we have both ancient and modern individuals from Andes forming another cluster. And then the Brazilian individuals again falling together. And then we have this Belize individual kind of in between. So what we try to do is like, okay, the ultimate goal of this exercise is see if we can reconstruct a kind of a tree, a graph that can represent all these individuals together in a phylogenetic manner. And so we start to kind of build this so-called QP graph of early Americans. And after the formation of the source population of all Native Americans, that, as I told you before, is a mix between Han Chinese and these Siberian ancient hunter-gatherer. There is a first split of ancient Beringian, a genome found in Alaska. And then, again, this really quick diversification that happened probably around, before 15,000 years ago in North America between this ancestral B and ancestral A. And what was really interesting to see is that within the ancestral A lineage, all the diversity of the ancient Central and South America fall. So all ancient Central and South America are part of this ancestral A lineage. And what you could notice really clearly is that a genome from different locations, from the Andes, from the Southern Cone, from Belize, and from Brazil, they kind of diversify extremely fast with a sort of star-like diversification. Meaning that, or we could speculate that really people arrived in South America and spread and diversified extremely fast, so that they are different branches on a star-like diversification. The problem starts, and it becomes much more complicated when we include in this admixture graph Clovis, when we add the oldest genome from North America. And so what we see, when we do this exercise and we add Clovis, ANSIC1, we see that it becomes like a spaghetti kind of tree. But there are certain characteristics that they're shared with the previous graph. That is, of course, this diversification, early diversification between ancestral B and ancestral A. There is, again, this star-like radiation of South American individuals. But what is interesting, we identify two additional gene flow from North to South America. One gene flow is this one in light blue, is a gene flow of a specific affinity from Clovis into the oldest genome from South America that are, as I told you before, Chiles losriles 11,000 years ago, as well as Lapadosanto 10,000 years ago. But this is not the only signal that we find, not the only gene flow that we find. There is an additional one that appears much later in individuals from Peru, at least from 4,200 years ago, from a group related to the California Channel Islands. So if we try to sum up a little bit what I just showed you before in a kind of graphical abstract, we identified a total of 4 genetics exchanges between North and South America. One is a primary source of ancestral ancestry in all South Americans, at least by 9,000 years ago. Then you have again a new lineage with distinct affinity to ANSIC1, and that we find in the oldest individuals from Lapadosanto and Los Riles, and then yet a third gene flow event that happens much later, around at least by 4,200 years ago, and we link to population from the California Channel Islands. And of course, the population Y signal is found in modern-day Amazonians that we could not find in any of our ancient samples. But of course, as you can see, we don't have samples from Amazonia. So I will skip on these slides just to tell you that we are doing much more work now on the Caribbean, but I'm happy to talk with you about that later. And I would like to just conclude with these last slides about what are the next steps. And we finished the paper in 2018 by writing from the period between 11,000 and 3,000, that includes most of our individuals. We lacked ancient DNA data from Amazonia, North and South America, and the Caribbean, and thus we cannot determine how individuals from this region relate to the ones we analyzed. So what we're actually doing today is try to expand the collection of ancient genomes from Northwest and South America. We are really interested also to get more data from the Circum-Caribbean region. And of course, one of the reasons why we are here, I'm really interested to talk to you, is also establish collaboration with Cuban researchers that share the same interest, and potentially we can study the population history of Colombia and the South American together. And with that, I would like to acknowledge the group in Jena, and especially a PhD student, Catherine Negele, that is actually worked a lot in this, on this topic, and you for your attention.